Through investigating the genome sequence, I found it significant that the S protein of the Wuhan coronavirus was critical in its cross-species ability to infect humans. While I was searching for related studies online, one Chinese virologist in particular caught my attention. She spent many years researching bats and coronaviruses. She was the first to locate the key to how coronaviruses can overcome cross-species barriers in order to directly infect human bodies. And she was the first to discover that the SARS virus was the result of a restructuring of multiple SARS-like coronaviruses found in bats. Her name is Shi Zhengli, and she may be an important link to the origin of the virus. Wikipedia describes Shi Zhengli as a, quote, Chinese virologist and researcher at the Wuhan Institute of Virology, which is part of the Chinese Academy of Sciences. Further investigations show that Shi Zhengli has been a figure of controversy since the Wuhan virus outbreak. This is due to a paper she published in 2015 discussing her own research into synthetic viruses.
Dr. Zhengli, she is one of the top experts in China studying about coronavirus in Wuhan Institute of Virology. She has so many publications from collecting, identify bat coronavirus from bat caves. Her lab has these capacities and very sophisticated capacity to generate mutations to make it best fit in human expression as well.
Delving further into related information, I discovered that Shi Zhengli published not one, but 4 papers in total, each of which contains important information. Since the SARS outbreak in 2003, Shi Zhengli has been conducting research on coronaviruses. From 2010 onward, the focus of Shi and her team was redirected to identifying the capacity for coronavirus transmission across species, specifically putting the spotlight on the S protein of the coronaviruses. In other words, her team's research in the Wuhan lab has been looking into the part that can make coronaviruses transmittable to humans. In June 2010, a team including Shi Zhengli published a paper. It describes research to understand the susceptibility of angiotensin-converting enzyme 2, ACE2 proteins of different bat species to the S protein of the SARS virus. In the experiments, they also modified key amino acid convents to mutate the bat's ACE2 to examine compatibility with the SARS S protein. This paper demonstrated their awareness of the special relationship between the S protein and the ACE2 receptor. It also signified that Shi had unearthed the passageway for coronaviruses into human bodies. In October 2013, Shi and her team published a paper in the authoritative science journal Nature. They claimed a breakthrough in coronavirus research. What was their breakthrough? They successfully isolated 3 viruses from bats, one of which had an S protein that integrated with human ACE2 receptors. This effectively demonstrated the human infection of SARS-like viruses to humans without the need of an intermediary host. Then, in November 2015, she and her team at the Wuhan lab once again published a paper, this time in the British journal Nature Medicine. They discussed the creation of a synthetic virus, a self-replicating chimeric virus. This virus had the SARS virus as the framework, with the key S protein replaced by the one they had found in a bat coronavirus she mentioned in her 2013 paper. This new virus demonstrated a powerful ability for cross-species infection. The mice infected with this synthetic virus revealed severe lung damage with no cure. This symbolized that Shi's successful splicing of the SARS virus was a key to open the door to the cross-species transmission. What is startling is that these successful experiments on mice were only the tip of the iceberg. They planned to further experiment on primates. Although Shi Zhengli did not indicate any conclusion from this research, her move to research on primates suggests this was to more closely simulate the infection of humans with this new synthetic virus. This wasn't done without controversy, however. Xi's experiments quickly triggered widespread debates from the academic community. Simon Wayne Hobson of the Pasteur Institute in France expressed deep concerns. He told Nature, if the new virus escaped, nobody could predict the trajectory. Propagation could happen anywhere.
His statement is exactly what's happening, that the virus is everywhere.
However, this did not terminate Xi's research. On November 14, 2018, Xi Zhengli spoke at the School of Life Sciences and Biotechnology at Shanghai Jiaotong University. The topic was bat coronavirus and its cross-species infection. Reports of this event have since been deleted from the university website. I discovered two more significant pieces of information regarding the dangers of the research conducted by Xi Zhengli's team. First, on October 16, 2014, the Obama administration, wary of the potential threats to public health from the gain-of-function GOF research into SARS, MERS, or influenza, announced through the National Institute of Health that it was suspending funding into similar research. The funding pause included Xi Zhengli's research project, Genetic Engineering of SARS-like Coronavirus in Bats, a collaborative effort with Dr. Ralph Barrick, a virologist at the University of North Carolina. Second, after the Wuhan outbreak, Indian researchers compared the S protein sequence between 2019 nCoV and SARS. They discovered that 2019 nCoV had 4 new sequences inserted, all of which can be found in HIV sequences through a search on GenBank. Xi Zhengli discredited those observations, although she never denied the existence of the 4 inserted sequences. However, scientists probing GenBank found that there were only 3 viruses containing all sequences. The first is the HIV virus itself. The second is a bat coronavirus discovered by Xi. And the third is this new Wuhan coronavirus.
We've done this kind of work for now 40 years for me. There's the sequence analysis and comparison of the virus of the SARS-2 COVID-19 apparently has genes that come from other human and other species, including some envelope, the GP41 from HIV.
What is the HIV's GP41? The answer I found online describes GP41 as a protein of HIV that acts as the key to infecting human bodies, resulting in the functional failure of the immune system. If the discovery by Judy and her colleagues are established, it would mean that the infectious part of the Wuhan virus, the S protein, incorporated the sequence of the HIV key protein. This made me think of the immunodeficiency symptoms in people infected.
They were doing research on a human transmittable coronavirus that was actually published in a paper. So this is research that they actually published. They were working on developing a coronavirus for the human host, which, leads you to question, why would you be creating a coronavirus that can infect humans? What would be the purpose of that research? Is it for a weapon? Is it so that you can then create a vaccine that you are the sole recipient of the profits from? The Chinese have full access to our databases. They have full access to all that research that comes out. They have full access to all our universities to train their scientists. And they have full access to our scientists, like was, with the recent indictment of the head of the chemistry department at Harvard. I mean, this is the 1,000 Talents program. Tens of thousands of the world's most brightest people in all of these different areas that are going to China to help them with their programs. And all of these programs, as you know, have a dual-use capability.
January 23rd, 2020. The Wuhan virus exploded. While Wuhan announced the lockdown of the city, Xi Zhengli and her team released a paper stating that the Wuhan coronavirus was of probable bat origin. This was published in Nature on February 3rd. The paper indicated that the Wuhan virus utilized the same key as SARS to gain entry into the human body. She also announced the 2019 nCoV genome sequence was 96.2% consistent with a bat coronavirus originating in Yunnan, China, called RATG13, signaling a natural source of the Wuhan virus. However, Shi Zhengli's natural origin assertion was doubtful. The outbreak occurred in Wuhan, the same location as the P4 laboratory where Shi was based, and which housed highly similar viruses. Common sense would lead the government to first inspect the P4 laboratory for any leakage incident and potential safety concerns. Instead, they shifted public attention from the P4 laboratory to the South China seafood wholesale market that sold no bats, and designated it as the origin of the disease. At the same time, authorities sealed off all virus samples, prevented international experts from joining the investigation, and used national television to slander doctors such as Li Wenliang, who disclosed the outbreak for spreading rumors. If the Wuhan virus indeed emerged naturally, why would the CCP need to censor relevant news or block investigations? Could the Wuhan P4 laboratory have its secrets? Virus samples and genome sequences may be the exact ingredients we need to find our answers. January 2nd, an email from the Director General of the Institute to all internal staff was circulated. The subject was Notice regarding the strict prohibition of disclosure of any information related to the Wuhan unknown pneumonia. National Health Commission clearly mandates that all detection, empirical data, results, and conclusions related to this outbreak cannot be published on self-media or social media, nor disclosed to any media, including state media, or collaborative organizations, including any technical services companies. January 21st, a new drug, remdesivir, provided for free by the United States to China for Wuhan coronavirus treatment, was preemptively patented by the Institute. February 3rd, Dr. Wu Xiaohua blew the whistle using his real name that Shi Zhengli's haphazard laboratory management may have led the Wuhan virus to leak from the lab. February 4th, Chairman of Duoyi, Xu Bo, blew the whistle using his real name that the Wuhan Institute of Virology was suspected of manufacturing and leaking the Wuhan virus. February 7th, top biochemical weapon expert of the People's Liberation Army, Chen Wei, officially assumed control over Wuhan Institute of Virology's P4 laboratory. February 14, Chinese leader Xi Jinping called for the inclusion of biosecurity into China's national security framework and to accelerate the introduction of a biosecurity law. February 15, the Institute refuted widely spread rumors on Chinese social media that a female graduate, Huang Yanling, was patient zero and had perished. However, Huang's photo, CV, and thesis were all removed from the Institute's official website, leaving only her name. February 17, Institute researcher Chen Chuanjiao blew the whistle using her real name that Director General of the Institute, Wang Yanyi, was suspected of leaking the virus. Military biochemical weapons expert assumes control. Biosecurity law. Now, when these sensitive keywords are connected together, it tells me that the P4 laboratory is not an ordinary academic research institute. I decided to start from the beginning of the Wuhan P4 lab. On January 23rd, the day Wuhan was locked down, French website challenges.fr published an article that revealed many details of the collaboration between France and China to establish the P4 laboratory in Wuhan. In 2003, after the SARS outbreak in China, the Chinese Academy of Sciences requested assistance from the French government to build a virology research center of the highest level. With the support of then French Prime Minister Raffarin, the two countries signed a contract to jointly construct the P4 laboratory under a wave of contention. According to the contract, French architect RTV in Lyon was responsible for the engineering of the laboratory. However, Chinese authorities switched the work to a local Wuhan architect, IPPR, based on research conducted by the General Directorate of External Security. IPPR had close ties to subsidies of the Chinese military, some of which were already targets of concern by the American CIA. Within the 12 subsidiary departments, there was even a specific military management office. The French delayed repeatedly, given security concerns. It wasn't until 2017 before the Wuhan P4 laboratory was operational, and French security continued to suspect that the Chinese regime was conducting biochemical weapons experiments. Who is the real boss of the P4 laboratory? The Wuhan P4 laboratory was a subsidiary of the Wuhan Institute of Virology, which is managed by China Academy of Sciences. The director of the laboratory was Yuan Shiming, who was also the head of China's Academy of Sciences Wuhan chapter. Design and funding for construction were the responsibilities of the ex-vice presidents of the academy, Jiang Mianheng from 1999 to 2011, and Chen Zhu from 2000 to 2007. Jiang Mianheng was the eldest son of ex-CCP leader Jiang Zemin. After Jiang Zemin ascended to power after the Tiananmen Square massacre, his son entered the academy and led the Institute of High Technologies Research and Development. Jiang Mianheng created the Shanghai Institute of Life Sciences, and together with China Academy of Sciences, Shanghai colleges and universities, Shanghai hospitals, military hospitals, and research institutions, formed a profit group of life sciences organizations. They controlled China's major life sciences research projects and allocation of massive funding. Jiang Jicheng, son of Jiang Mianheng, is the controlling shareholder of Wuxi AppTec, which in turn is controlling Fosun Pharmaceutical, China's agent for Remdesivir. Effectively, Jiang Jicheng is the kingpin behind the specific medicine for the outbreak. Meanwhile, Chen Zhu is the current president of the Red Cross Society of China, which had faced numerous scandals since the outbreak. In 1999, while Jiang Zemin was in power, the People's Liberation Army published a book, Unrestricted Warfare, in which strategies for a weaker nation to combat a stronger nation are discussed in the context of modern warfare. One of the authors, Qiao Liang, wrote, "After the first Taiwan Strait crisis, we realized that if the Chinese and American military fought head-on, we are at a disadvantage. Therefore, we need a new strategy to help our military tilt the balance of power. This new strategy is called unrestricted warfare, a variety of means beyond all limits, without any restrictions. It could be military related, including guerrilla warfare, terrorism, biochemical warfare, or it could be non-military related, such as drug trafficking, poisoning, environmental destruction, and computer virus dissemination." Israeli expert in biological warfare and former intelligence officer Danny Shoham published a paper in 2015 in India's Journal of Defense Studies. He pointed out that China will not miss, skip, or abandon any highly advanced technology, especially military-related technology. The article noted that China had 12 facilities related to the Ministry of National Defense and 30 subsidiary facilities of the PLA participating in the research, development, manufacturing, testing, or storage of biological weapons. The Federation of American Scientists indicated similar concerns in an evaluation report. The organization believed that the CCP possessed advanced chemical warfare projects, including research, development, manufacturing, and weaponizing capabilities. The CCP is usually known to have a proactive biological warfare portfolio, including state-funded and sponsored research activities. This virus must have a message for all of us as people of this earth. We need to take a break from our busy schedules and seriously consider the repercussions of our ever-expanding scientific research and development. Did we go too far? Do our desires and ambitions go against the laws of nature and pull us further from the direction given by heaven? Maybe when this virus lifts, we can again find our traditional values, rediscover kindness and virtue, family and community. I believe that viruses can't survive where hearts have compassion.